But you could take those, and one of the things that we could do, they were very, very highly uneven, so we ended up with, just from a very small library, basically, we were able to completely assemble the genome of 3 viruses, for which, of course, we don't know the hosts, but they're quite interesting, and so 2 of them are picornolite viruses, and the third one is a distant relative of Umbra and Tombus viruses, which are viruses which infect higher plants. And so I'm really convinced that this is the origin of all of these RNA viruses which infect higher organisms. And you can see, you can kind of look at the DNA in these things and recognize that they kind of in some ways look a little bit primitive relative to other things. And then you can do phylogenetic analysis. This is the RNA-dependent RNA polymerase of the Picornovirales. And I don't know if I have a pointer here or not, so I'm not going to try it or it will fall apart. And the batteries just fell out, which is not a good sign. So, so you can do these phylogenetic analysis, and if you do it on this whole group in the bottom, are the things that we pulled out of our metagenome, for example. The 2 things, the little red circles, JPA and JPB, are the complete viral genomes that we sequence. Harn of the blue one is this isolate that we've got. And so you can see this big cluster of diversity. So in 2 water samples, basically, we came up with about another 4 or 5 putative new families of Picornovirales. So the diversity is extremely high. If we go and take a look at something else, so this is sort of a metagenomic analysis take-home message then that most of these viral sequences that we pull out have no recognizable homologs in databases. And so we could ask the question, well, what if we kind of if we go in and we look at specific groups of viruses and look at the diversity within those, or how much genetic variation there is. And I'm going to talk about some phage at first. I'm going to talk about potoviruses and myoviruses. So potoviruses are your T7-like things that probably most of us have heard of at some point. And then the myoviruses are these guys like T4, right? So with the big contractile tails. And so if we look at potoviruses, and here's some samples. This is some work by Karen Reed and Jessica Labonte, 2 graduate students in my lab. And so we took some samples again off the coast of British Columbia. And if you start doing phylogenetic analysis on the DNA polymerase of these, you can build a big tree of the recognizable, essentially, T7-like viruses. And the interesting thing is that those 3 big phylogenetic groups were completely unknown until we took those 3 water samples, right? So these are a pretty heavily studied group of phage, T7-like phage. And so the diversity, again, that we're finding, so these are groups that have no cultured representatives, but clearly are quite divergent. You can do the same thing with T4-like viruses. Again, here's where we collected our samples from. And you can, again, build a big tree. This is some work we did with Henry Krish and Jonathan Filet of CNRS. And what we can do is we can see, it's quite interesting. You look at these big groups. In this case, this is the major capsid protein. And you can see there's actually biogeography here. You can see the blue ones are from the Pacific Ocean, the green ones are from the Arctic Ocean, the red ones are from the Gulf of Mexico, and you can kind of see them sorting themselves out a little bit. And so again, the cultured representatives are the ones that are in black, and there's not very many. In fact, all of those are completely new groups of T4-like phage, and Henry Krish was all excited about this, and I said, well, that's what we always find. And he said, no, this is big news around the people that work with T4-like viruses. So again, we see this massive diversity. We see some interesting patterns of biogeography, and we're kind of trying to tackle some of those issues as well. So the question that remains is, so what about the frequency distribution? So we see there's all this diversity, are these populations relatively uneven or not? So we took what we call our GOB project for global ocean biodiversity, and we took 3 water samples around Vancouver again, and we looked at a whole bunch of different virus groups by essentially deep sequencing with 454 again to look at specific genes within specific groups of viruses. And I'm only going to talk just about 2 ones very briefly here. But if you look at myoviruses again, so these are the G23. So this is a frequency histogram. So on the y-axis, the vertical axis, is the number of events that occurred, and on the x-axis is a particular OTU, or a particular gene sequence for that virus. And so what you can see is that they're highly uneven, so that we have a few viruses where that gene sequence appears quite often. But the amazing thing is that 90%, even after 1,500 sequences, 90% of the time we'll pull out a sequence we've never seen before. Okay, from this one gene for this one kind of small group of viruses. So that's from GOB1. If we look at GOB2, the other one, again, the same thing. This case, about 1,000 reads. Again, about 90% are singletons. We're only seen once. And if we look at GOB3, again, the pattern's the same. Again, about 1,400 reads here. and almost all of them singleton. So you see this massive sort of unevenness of the population. And then you can ask the question, do the same OTUs, the ones that are dominant in one location, are they the dominants in the other location? And in fact, what you see is, well, it's kind of variable. So if you take the most dominant genotype, you can see, which is on the far left side there, you can see that it occurred in all 3 locations. But if you look at the second most abundant genotype, it only occurred in 2 out of those 3 locations. And so you can see as you go along that pattern here, again, it's quite unpredictable in terms of whether you're likely to find the same genotype at all 3 locations. So we can do another thing, here's another one, phycodinoviruses, which are big double-stranded DNA viruses related to pox viruses, iridoviruses, baculoviruses, and Mimi virus, for example, and ask the same kind of question. And again, if you look in the ocean again, I think what's quite remarkable here is the yellow ones are the cultured representatives. Everything else, this is the DNA polymerase gene, represents the big family of these viruses. So you can see that we have very, very little of the diversity in culture. But you can ask, again, you can go out there and look at the frequency distribution and of DNA polymerase sequences, and again, you see this same pattern. In this case, there was only 184 reads, but again, almost 90% of them were singletons. So you see that, again, with these very, very uneven populations. So the final point then is, I hope I've convinced you that the diversity of viruses in the ocean is enormous, but largely unknown. I only focused on a couple of groups. We've looked at lots of different groups of viruses. And that there's this vast reservoir of unexplored diversity that we still have to go after. And so maybe we'll go after retroviruses next. So the take-home point, only 3 things. There's about 75 million blue whales worth of carbon in viruses in the sea, about 50 million per mil. There are major agents of mortality in biogeochemical cycling, and there's this enormous diversity. And finally, I'd just like to thank the folks in my lab that make all this possible. Thanks very much.
Time for a couple of